Assalamu alaikum dear students, hope all of you will be fine. In our today's lecture, we will describe a short content so far as our the lectures which we are delivering on the process of digestion in the physiology and anatomical chapter of your syllabus. And in our today's lecture, we will briefly describe the composition of the bile. Then if we will get some time, then we will also briefly describe the composition of the pancreatic juice. Then in our coming lectures, we will be describing their formation as well as their role for the process of the digestion in our digestive tract. And I have made a simple diagram here in which I have shown that there are two important these accessory digestive glands in our digestive system. One among that is liver and another one is that of our pancreas. Since you know all of the anatomical structures, their location at histological level even, but in our today's lecture, this simple diagram will simplify or exemplify us that where from the bile comes and what are the different tracts through which the bile reaches up to our duodenum, and where from the pancreatic juice comes and what are the different tracts through which that pancreatic juice is transported and is being incorporated into our duodenum. And so far as our previous lecture is concerned, in which we discussed how the emptying of the stomach took place. Once the emptying of the stomach starts, then there is the beginning of the inculcation of what? This time into our duodenum. And so far as the whole process of the digestion in our intestinal part of the gastrointestinal tract is concerned, this is that very main structure, what is known as the duodenum, which is of the utmost importance, without which the process of completion of the digestion of the fats, the digestion of the lipids, the digestion of these nucleic acids and most of the other biomolecules is not possible for a human being. So, now let us have a simple look over this very simple diagram in which it is clearly mentioned that here it is our liver and this very bag like structure is our gallbladder, you already know it, and the duct which is draining your gallbladder towards the exterior side, that very duct is known as cystic duct, and from the upper right side, from the upper left side of your liver, there is a beginning or there is presence of a duct which is draining the bile from that very side known as left hepatic duct, and this very duct which is here, that is here this very duct. I have made this line here, actually that is the line which comes from right hepatic duct, and this very line is indicating this duct which is the cystic duct which is draining the gallbladder. And when these two right and the left hepatic ducts get combined with one another, they get merged and form a common duct, what is known as common hepatic duct, and common hepatic duct gets merged with the cystic duct to form a very common and large duct, what is known as common bile duct, and then common bile duct is finally almost gets fused with another duct which is draining the pancreas, and that very duct is known as pancreatic duct. When both pancreatic duct and when pancreatic duct will be combined with the common bile duct, then ultimately this in this very region, very smaller portion in which there will be transportation of both the pancreatic juice as well as the bile, that very part, that very region of this common duct will be called as hepatopancreatic duct. Besides that, we are having some other ducts which are collectively called as accessory pancreatic or accessory pancreatic bile ducts, APDs, accessory pancreatic ducts. This is the duct system. See, this very side of the liver will be drained by left hepatic duct, it will be drained by right, then this one will be drained by the cystic duct, and ultimately they will be drained collectively by common bile ducts, and this very region of the pancreas will be drained by, it is one of the major interior duct, what is known as pancreatic duct, which gets fused with the common bile duct to form hepatopancreatic duct here, and hepatopancreatic duct then gets incorporated into a ampullary region of our duodenum. That very ampulla is known as ampulla of Vater, and towards the interior side of the ampulla of the Vater, there is an important sphincter which is guarding and regulating the entry of pancreatic juice as well as the bile into the duodenum. That very sphincter is called as sphincter of OD, isn't it? That very sphincter is called as sphincter of OD. Now, in the liver, where the metabolism of this heme along with the globin and some of the cholesterol, fatty acids and some other things takes place, that metabolism ultimately results in the formation of the bile. In the formation of the bile, there are many enzymes which are involved in the metabolism. that in detail will be discussed in some of our coming lectures, in which we will be discussing the histology of the liver thoroughly, and after that we will be discussing the metabolic pathways which are undergoing with respect to different biomolecules and different biochemical substances in our liver. But here let me state some of the few, let me write down some of the few important enzymes which are mandatory for the formation of the bile or its contents. So, one major important this enzyme is 7 alpha hydroxylase cytochrome type of the enzymes, and sometimes it is called 71, and another one is 12 alpha hydroxylase, and another important enzyme is alpha D hydroxylases. All these enzymes are mainly associated in one of the important content of our bile, that is a bad substance. Should I write down here? Cholesterol. Though cholesterol has its own importance so far as the maintenance of the blood pressure and the flexibility of the membranes are concerned, but this cholesterol sometimes may put at risk when its limit goes to the higher side. Then its increased level can lead to the different problematic conditions in our blood, in our liver, in our metabolic pathways, and so many other things which will be discussed in some other lectures. But here I have write down 7 alpha hydroxylase, 12 alpha hydroxylase, alpha D hydroxylases, which are involved with the degradation, with the metabolism of the cholesterol, and thereby it is conversion into the different contents which will be present in the bile. Now, if we will be having a thorough look over the composition of the bile, we will find there that bile consists of many substances, the important among which will be present in the majority, that will be water. So, bile will consist of water, and it will consist of bile pigments. It will be also consisting of bile salts, isn't it? Besides that, the bile will be also consisting of lecithin, cholesterol, and it will be also consisting of sodium, little bit chlorine, potassium, calcium. These are the ionic formatives which are present in your bile, and carbonate ions. So far as the composition of the bile is concerned, the main thing which plays an important role in the process of the digestion of especially the fats, they are these bile salts. And after that, we also have bile pigments which have their own importance for inculcation of the color to some of our things, some of our structures, some of our substances. So far as the common composition of the bile is concerned, you have seen that bile consists of water, it consists of bile salts, it consists of bilirubin, consists of cholesterol, it consists of fatty acids, it consists of lecithin. It will be containing sodium, it will be containing potassium, it will be containing this calcium, it will be containing chlorine, it will be containing this HCO3 negative ions, or the carbonate or the bicarbonate ions. And so far as the bile pigments are concerned, let us have a thorough look over the bile pigments. The important bile pigment which is present in our liver is known as bilivirdin. Important pigment is bilivirdin. Where from this bilivirdin comes, I will let you know. But so far as the color of the bilivirdin is concerned, it is usually green in color. Remember it. it is green in color, but in many of the animals including the humans, this green colored bilivirdin is being reduced in the liver into its another form known as bilirubin. And now the color of this very bilirubin is not green, it has now been changed and its color is now almost yellow. So bilirubin is a yellow colored bile pigment which is mostly present in our bile. See, actually the RBCs are taking almost 120 days to complete their life cycle. Everybody knows it. When the life cycle of the RBCs is completed, then it becomes dead. And you know that the main constituent unit which is present in the red blood corpuscle, that is hemoglobin. And that hemoglobin remains there within the Donane's membrane of the red blood corpuscle. And such red blood corpuscles are being thrown into their graveyards, and the graveyard of RBC is what? Your spleen. And most of the metabolism or the metabolic pathway for the degradation of this hemoglobin, the content of the RBC, is being performed by this very spleen. But here we are not discussing the whole pathway of that bilirubin which is taking place in the spleen or in the liver, but here we will only tell you that the red blood corpuscle which was containing what? Which was containing the hemoglobin. When it reaches into our liver, it is being degraded into two parts. Number one is heme and another one is globin. And you all are familiar about this very fact that the globin is actually a protein. If it will be degraded, its degradation will lead into the formation of the amino acids. Then these amino acids will be reabsorbed in the different regions or by the biliary system of our liver, or when if they will reach up to the intestines, they will be still absorbed. And after their absorption, they will be utilized for different purposes by different cells of our tissues. So we will leave this part, that is the globin. Then this heme will remain there. In this heme, there is one part, that is iron. This iron will be taken and it will be sent to the bone marrow, isn't it? It will be sent to our bone marrow, where it will be utilized by the pluripotent stem cells for resynthesis of the hemoglobin or the RBCs. But the remaining almost proteinaceous part of the heme, this very part, and some of the iron, it will now be utilized by the different enzymes in the metabolic pathways of our liver, and that very part will be finally converted into bilivirdin, and this bilivirdin will be then reduced into the bilirubin. And so far as this bilirubin is concerned, this bilirubin along with these all substances, they will result in the formation of the bile. Clear? And so far as this very bilirubin is concerned, it further is being reduced. It is further being reduced, and this bilirubin is being converted into the mesobilirubinogen. It is being converted into mesobilirubinogen. This is mesobilirubinogen. It is the reduced form of bilirubin. And this mesobilirubinogen can also and further be converted into the cetylchobilinogen. And cetylchobilinogen, isn't it? And so far as this cetylchobilinogen is concerned, including the mesobilirubinogen, this cetylchobilinogen will be oxidized into cetylchobilin, which is a brown pigment. This is oxidized into cetylchobilin, Ceticobilin. Remember that it is almost having brown color. Not only that, you have to remember one more thing which is lying associated with this, that is it is responsible for color of our feces. Color of feces. For the color of feces, it is the ceticobilin, which we have got because of some oxidative pathway being taken out by the different intestinal enzymes, and which we have got from the oxidation of ceticobilinogen, and ceticobilinogen we have got from mesobilirubinogen, and this we have got from the bilirubin, and this one we have got from bilivirdin, and bilivirdin we have got the heme part of our hemoglobin, isn't it? And sometimes this ceticobilinogen is also converted into, it is one of the another format which is known as urinobilinogen. Sometimes this very format is converted into urinobilinogen. As the name suggests, it will be responsible for the imparting color to urine. The color of our urine is because of urinobilinogen, but it has come from what? From the degradative metabolic pathways of our heme part of hemoglobin of our red blood corpuscles. Sometimes this very pigment is also known as urobilin. Sometimes it is also known as, you will remember here one more name, urobilin or urochrome. So urobilin or urinobilinogen is that very pigment which is found in our urine or responsible for the imparting of color to our urine, isn't it? And this very pigment as well as this very ceticobilin pigment, which are respectively responsible for the color of urine as well as our feces, both we get from the metabolic degradative pathways of the bilirubin, which we get from bilivirdin, or which we get from the heme part of our hemoglobin. And now, if we will be having a look over the salt composition of our bile, then our bile will be consisting of two important formats of the salt. Let me write down here. Bile salts. Bile salts are present mainly in two formats in our bile. Number one is sodium glycolate, and number second sodium taurcolates. When we will be discussing the thorough synthesis or the metabolic synthesis of the bile as well as its contents, there we will come to know that these two important salts which are primarily responsible for the lowering down the acidic or the acidic condition of the chyme. There we will come to know that the sodium glycolate as well as the sodium taurcholate are actually synthesized with the help of two important amino acids. One important amino acid is glycine and another important amino acid is cysteine. Okay, another one is cysteine. All of you know these two types of important enzymes. Then this cysteine, this glycine is converted into its another form, that is the glycol, and cysteine is converted into taurine. You will remember it, it's converted into taurine, isn't it? Then both of these will be combined with another substance, that is cholic acid. That is cholic acid. It will also be added with cholic acid. When this will be combined with cholic acid, finally it will result in the synthesis of, along with the sodium glycolate, which is very basic in nature, an important salt. And When taurine will be combined with what? Cholic acid, then both of them will be converted into the sodium taurcolate. In this way, these two important and many other types of the salts which are present in our bile are synthesized. When the bile is synthesized, then this bile, when we have empty stomach or when we are not eating things, when we are asleep, then this bile gets transported in these canals like there. When we are awake and when we are indulged with the process of the digestion, then this very pathway is stimulated, and the stimulation of the liver for the synthesis of the bile takes place under the influence of a mucosal secretion, which you know, which we discussed in our previous lecture, that was secretin, and it will be the secretin which will guide both. See, this is the secretin here, it will come along with the blood and it will induce it for having good synthesis of the bile, and it will also induce the pancreas for having a good synthesis of the pancreatic juice, which we will discuss now tomorrow. And after that, another hormone will be synthesized, which was called as CCK or what? Or cholecystokinin. Here this is CCK. The CCK will come and it will lead to relaxation of these very muscles which are draining, which are present in the ducts which are draining the gallbladder, and then these after the induction of this cholecystokinin, there will be release of bile as well as release of pancreatic juice, and this as well as there will be relaxation of the sphincter of the OD muscle, and ultimately there will be inculcation of this very bile along with these substances into the duodenum. And in the duodenum, which just has received the chyme, the acidity of that chyme will be neutralized under the influence of these bile salts and these ionic substances, isn't it? So, if we will be having a look over our today's small lecture, in which we discussed the composition of the bile, we said that the bile is the secretion being synthesized by the liver and stored mainly in the gallbladder, thereby it is being transported into the duodenum, and where it will play a major role for the digestive pathway, especially with respect to the lipids. And we came to know that the bile consists of mainly water, pigment, salts, lecithin, cholesterol, xenophobics, which we will discuss it in later of our lectures. And so far as the bile pigments are concerned, the main bile pigment was bilivirdin, which we got actually the degradative metabolism of our heme part of our hemoglobin, primarily in the spleen, then from in our liver, and then that bilivirdin was reduced into bilirubin, and which was reduced into the bilirubinogen, which was reduced into the citricobilinogen, and citricobilinogen was converted under the influence of some bacterial enzymes into the citricobilin, then which was responsible for imparting for inculcation of the color to our feces. And we also came to know that the urinobilinogen or the urobilin is also an outcome of such pigmented substances, which are then being absorbed. We will come to know about the absorption process in our later lectures. Which are then being absorbed, then which are being excreted out along with our urine, and which are responsible for imparting the color to our urine. And we also came to know that there are two important bile salts, that is sodium glycolate and taurcolates, and sodium glycolate has been synthesized because of the glycine of this glycol form along with the cholic acid, and the sodium taurcolate has been synthesized because of cysteine and amino acid in its taurine form along with the cholic acid. So, with this we have concluded our today's lecture. In our coming lecture, we will be having a thorough discussion over the composition of the pancreatic juice and the role being played by both bile as well as the pancreatic juice in the process of digestion. Till now, stay blessed. Thank you.